Good afternoon, everyone. So, till yesterday we have discussed about mitochondria and endoplasmic reticulum and what are the processes which takes place in them and how it looks like, what is the structure, and all these things we have discussed. So, today we will discuss about the other organelle which are important in the cell. So, the first one is lysosomes. So, lysosomes are membrane-bound organelle which are found in the cytoplasm of eukaryotic cells. So, previously, as I told you, these lysosomes consist of some digestive enzymes and they are mainly present in animal cells, even in the plant cells also. They perform some important role as a vacuole. Okay, so these digestive enzymes are important for digesting the during the turnover process, whatever the organelle is, or if any pathogen enters into the body. So all these components, when they have to get rid from our body, so these digestive enzymes plays an important role. So lysosomes were discovered by a Belgian cytologist, Christian de Duve, in 1955, and he named them as suicidal bags or suicidal sacs. That means they are mainly involved in the removal of the dead content or unnecessary matter. So therefore, it is nothing but suicide. There is no killing from outside. During a normal turnover process, the body itself is removing some cells or some components of the cells. Okay, so therefore, they are called as suicidal bags or suicide sacs, and they are most abundant in cells which are related with the enzymatic reactions. Like wherever more metabolic processes involving enzymatic reactions takes place, so in such cells we can see increased number of lysosomes. Examples are liver cells, pancreatic cells, kidney cells, spleen cells, leukocytes, macrophages, etc. So if you remember in previous classes when I was telling you, like the during immune reactions, if a pathogen enters into the body, so first of all, phagocytic cells, they come and they engulf this bacteria or pathogen, and these phagosomes will fuse with the lysosomes and they form phagolysosomes, and these enzymes in the lysosomes, they digest this pathogen which is present in the phagolysosome, and that pathogen will be removed from the body. So here the phagosomes, macrophages also can act as a phagosome. So like that, in cells where the requirement of enzymatic reactions is there we can see large amount of lysosomes. So what is the structure of lysosome? So lysosomes are membrane bound, as we have discussed. So they are surrounded by a single membrane with unique composition. So its composition is different, and the membrane consists of highly glycosylated lysosomal associated membrane proteins. So there are some proteins which are very specific to lysosomes. Hence they are called as lysosomal associated membrane proteins and lysosomal integral membrane proteins. So in plasma membrane also we see this membrane associated proteins or peripheral proteins and integral proteins. So integral proteins are nothing but which pass through the membrane, and peripheral proteins or membrane associated proteins, they are loosely attached to the membrane. So in the same way, here also in the lysosome, we can see transmembrane or integral membrane proteins as well as membrane associated proteins. But these are very specific to lysosomes. And around 50% of all lysosomal membrane proteins are made up of this associated membrane associated proteins and integral membrane proteins. So these two LAMPs and LIMPs, they form a coat on the inner surface of the membrane. So this protect the membrane from attack by a numerous hydrolytic enzymes. Okay, so which are retained within lysosomes. So what happens sometimes in lysosomes themselves contain some digestive enzymes which are hydrolytic in nature, means which cleaves some proteins or carbohydrates like this. So when such kind of enzymes are present, they can act on their own cell and they can destroy themselves. So in order to protect the membrane, these LAMPs and LIMPs will form a coat inside the surface of the membrane so that these hydrolytic enzymes cannot digest the or cannot damage the membranes. So the lysosomal membrane also harbor a proton pump that utilizes the energy for ATP hydrolysis to pump protons into the lumen, thereby creating acidic pH, which is characteristic of lysosomes. So yesterday when we discussed about mitochondria, we have discussed a lot about proton pumps, so how ATP is synthesized, all these things. So here also in the lysosomal membrane, we have a proton pump. So from ATP hydrolysis, whenever ATP is hydrolyzed, here it is not synthesizing something. When ATP is hydrolyzed to form ADP and PI, that energy, whatever the energy which is released, that energy is utilized by this membrane, that means the proton pump, to pump protons, H plus ions, into the lumen of the cell, lumen of the lysosome. Thereby, what happens? When more H plus ions are there, acidity, that is acidic pH, will be generated. So therefore, lysosomes are mostly acidic in nature. So the primary function of lysosomes is degradation of extra and intracellular material. So that's what we were discussing. So extracellular material means which is coming from outside, like pathogens or any dust material or during allergic processes. Whereas intracellular material means the cells or components of the cells which they have to be removed on a regular basis. For example, blood cells. After every 20 days, they have to be removed. Then new cells will be formed. So like that, every cell will have certain lifespan. Some may get damaged during some injury or something like that. So whatever it is, so when it has to be degraded, the lysosomes plays an important role. So for this purpose, lysosomes are filled with hydrolases. Hydrolases are the enzymes. So here 40 varieties of enzymes are present. So mainly these are classified into 6 main types. Proteases, which digest proteins. Then lipases, which digest lipids. Amylases, which digest carbohydrates, especially amylases sugars. Then nucleases, which digest nucleic acids. Phosphoric acid, monoesters. So these are the different forms of enzymes which are present. And the lysosomal enzymes are collectively called as hydrolases. So whatever it is, lipase or protease or amylase or whatever it is, they are together called as hydrolases. So they cleave the substrate by the addition of water molecule. So hydro means water. Hydrolases means whenever the product is formed, this water molecule will be cleaved and one molecule will get H and other will get OH. So that means with the addition of water molecules, the cleavage will take place. And as I told you, lysosomes are acidic in their nature. So therefore, the enzymes also function in acidic media. Hence, they are called as acid hydrolases. So this is the lysosome structure, how it looks like. It is a single wall membrane, and these are the inside are the enzyme complexes. So how these lysosomes are produced? So they are manufactured and budded into the cytoplasm by Golgi apparatus with enzymes inside. So they are like a vacuoles only. They are formed like a vacuoles through budding process. Budding is nothing but just enlargement of a component. Even in bacteria also we see budding. So when a cell or organelle it enlarges, then it folds inward from at the center, and now it will separate into two small parts. Okay, now each one can individually perform their functions, and the enzymes that are within the lysosomes are made in the rough endoplasmic reticulum. So usually lysosomes, they are budded off from the Golgi apparatus, and the enzymes which are present here, enzymes are proteins, right? So the proteins will be synthesized in ribosomes. So here they are made in rough endoplasmic reticulum. That means the ribosomes which are associated with the endoplasmic reticulum. Once they are synthesized, they are delivered to the Golgi apparatus via transport vesicles, and from the Golgi apparatus they are budded off. So like this. So from the Golgi apparatus, the primary lysosomes will be formed. Then they will fuse, different lysosomes will fuse and form secondary lysosomes. So what are primary and secondary lysosomes? So primary lysosomes are small sac-like structures enclosing the enzymes which are made from the rough endoplasmic reticulum. So that means they store these enzymes, and we can call them as granules, storage granules. Whereas secondary lysosomes, I told you, like phagosomes will fuse with the lysosomes. So this primary lysosomes, they fuse with phagosomes, then they form phagolysosomes. So they are actually secondary lysosomes. So secondary lysosome contains engulfed material and enzymes. And the materials are progressively digested by the enzymes in the digestive vacuole. So this is the primary lysosome which is having the enzyme. And this is the organelle. And this is autophagic vacuole. So the vacuole which is having this organelle which has to be digested. Now this will fuse with the primary lysosome to form secondary lysosome. Now here you have enzymes surrounding, and inside you have an organelle. So these enzymes will digest this organelle and we will get the residual body. So the main functions are heterophagy, programmed cell death, that is apoptosis. Autophagy means with self-eating, autolysis, fertilization, and chromosomal damage. So in all these things, lysosome plays an important role. So what is heterophagy? Heterophagy is the lysosomal digestion of extracellular material by process of endocytosis. So it is not an autophagy. Auto means self-eating. Heterophagy means, so the extracellular material gets digested in the lysosomes like pathogens by the process of endocytosis. So I told you, like endocytosis, pinocytosis, phagocytosis, in some other class I have discussed. So endocytosis means engulfing. They will engulf, the cells will take up the material, and this heterophagy is of two types: phagocytosis and pinocytosis. So phago means eating, cytosis means cell eating. Pinocytosis means, pino means drinking, cell drinking. If it is a solid material, so it is digested by the phagocytosis. If it is a liquid material or cytoplasmic contents or something, it is pinocytosis. Then programmed cell death or apoptosis. So here there are high levels of hydrolytic enzymes are present in the lysosomes. Therefore, they are harmful to the cell, because if it releases these enzymes, and it can degrade that cell itself, right? So partial and selective Lysosomal membrane permeabilization induces controlled cell death. So what happens whenever a cell is signaled to the death? Like I told you, the plasma membranes consists of inner leaflet and outer leaflet. So inner leaflet consists of phosphatidylserine. Whenever this phosphatidylserine, it flips to the outer leaflet, it gives a death signal, that is the apoptotic signal. So cell has to undergo programmed cell death. So during this process, how the cell undergoes death? This is by the lysosome, which is releasing some enzymes so that it slowly causes the death of the cell. So by how it releases? By selective permeabilization of the lysosomal membrane. So it provokes the translocation of lysosomal contents to the cytoplasm. Slowly it releases some of the enzymes to the cytoplasm so that it slowly degrades the cell. And the proteases implicated in the cell death are cathepsins. So these are the proteases. They remain active at neutral pH. So examples are cathepsin B, cathepsin C, cathepsin D, cathepsin L. They trigger and cascade the essential organelle of cell, which finally lead to the cell death. Then autophagy. So autophagy is nothing but self-eating. Auto means self. So autophagy is a normal physiological process in the body that deals with the destruction of cells. So it maintains homeostasis or normal functioning by protein degradation and turnover of destroyed cell organelle for new cell formation. So it's a part of homeostasis. So what is homeostasis? Means maintaining the cell, that is maintaining the balance in the body. So that homeostasis has to be maintained and normal functioning has to be done so that the proteins and all the other organelles or components has to be destroyed so that the new cells and new components will be formed. So it's a normal physiological process. There is no pathogenesis associated with it. So this next is autolysis. It is digestion of own cells by lysosomes. So in the autolysis, lysosomes digest its own cell and it occurs during amphibian metamorphosis and insect metamorphosis. That means, for example, a tadpole will become a frog. So if you say the structure, this is completely different and that is completely different. So the cells to get a shape, okay, or to change from one form to other, or caterpillar to butterfly. So that change in the structure has to be initiated by the autolysis. It will lyse some cells to get the structure. So here you can see like tadpole to frog, as I told you. So the structure is completely different. So to get a shape, these lysosomes will help in the autolysis. Then fertilization process. So during fertilization, the acrosome is nothing but in the sperm cell, in the head of the sperm cell, there will be acrosome, which is a lysosome, modified lysosome. So whenever the sperm fuses with the zona pellucida, a layer on the egg cell, so this has to digest that membrane and the components in the head should be released into the egg. So during fertilization process, this acrosome of sperm head, it ruptures and releases the enzymes. And these enzymes now goes and digest the egg membrane, that is zona pellucida, and it provides a way for the entry of sperm nucleus into the egg. Okay, so that the fusion and fertilization takes place and zygote is formed. So this is how it looks like. So this is the egg cell and these green ones are the sperms. So here in the egg cell, So this is the egg cytoplasm, and here we have a jelly coat, which is zona pellucida. Then you have a membrane. Okay, perivitellate space, cortical granules are present. And here, whenever a sperm has to enter into the egg, first of all, the mitochondrial material, nucleus, actin, acrosomal granules, all these are present. So this head group has to fuse with this jelly coat, that is zona pellucida. So here we have acrosomal granule, this blue color one. So that releases the components or enzymes which will digest this zona pellucida, and it will help in the penetration of the sperm. So finally here the head group is finally in contact with the egg cell, and it releases all its components. That means especially nucleic acid will be released into the egg cell, and this nucleic acid will fuses with the nucleic acid of the egg, and zygote is formed. Then chromosomal damage. So due to the presence of DNase enzyme, lysosome had an ability to attack chromosomes and cause chromosomal breakages. Okay, so they can lead to some diseases like cancer. So what happens is DNase enzymes which are present in the lysosomes, they can attack the chromosomes and can cause breakages in the chromosome. It is like a mutation. So this can cause some serious diseases like cancers and also some inherent diseases. Okay, so this is about the lysosomes. I will share you another.
Ma'am,
Yeah.
the matter which is given in the textbook
Yeah.
and the slides which you have presented is a little bit of difference, no, ma'am. So can we write even this in the examination?
Yeah, you can
The
write.
PPT notes which we
Yeah,
have taken.
whatever material you can write.
Because
Yeah.
in the textbook we don't have about the details about the enzymes and heterophagy and everything.
Okay. Yeah. 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 Because see, it is a vast subject. No book can cover all the topics. So yeah, textbooks you can keep it as a reference,
Okay,
and you
ma'am.
can take information. Everything is related to that only. So
Okay, ma'am.
even you cannot represent the entire topics in a single this thing, right? So
Okay.
whatever you remember, you just follow both so that whatever you remember, the important points you can cover.
Thank you, ma'am.
Okay. In textbooks, it will be in elaborated form. So I'm just telling you in a simple way. You can just add some points to this if you want, and you can represent in the examination. Okay.
Thank you, ma'am.
Okay. Now we discuss about, yeah. So in the next topic is ribosomes. Okay. So what are ribosomes? So ribosomes are the granular structures. Okay. So these are the mainly protein synthesizing machinery, and they are ribonucleoprotein particles. That means ribonucleus, that is RNA, what we call. Okay. So rRNA or ribonucleoproteins are present. So along with this, you have the protein portion also. And here, so the mRNA is acting as a blueprint for the process of protein synthesis. So we have DNA replication, then transcription, translation. These are all called as molecular dogma, okay, of cell biology or molecular biology, central dogma of molecular biology. It is nothing but how actually the gene expression takes place. So first the DNA will get replicated, and this DNA will code for mRNA during the process of transcription, and this mRNA will code for protein in the process of translation. So how this DNA is expressing in the form of protein, and once it expresses in the form of protein, then only we can get like a phenotype. Phenotype is nothing but a character. Okay, like for example, straight hair or curly hair, or like dark skin or fair skin. So whatever it is, whatever the character. So it is because of proteins. And that proteins information is stored in the DNA. But actually the information of protein is stored, it is coming from the mRNA. mRNA is a blueprint for the protein synthesis. So Palade, he was the first person to study ribosomes in 1955. He was the scientist. And so how many number of ribosomes are present? So the number differs greatly. So it is not a fixed number like only this much should be present, not like that. So E. coli cell may have up to 15,000 to 20,000 ribosomes, which are contributing to about 15% of the cell mass. So how they look like this, you can see small beads like this. And what are the different types of ribosomes? So there are matrix ribosomes, plasma membrane ribosomes. So matrix ribosomes means which are remain in the cell. So that means the cytoplasmic ribosomes, and they synthesize the proteins which remain in the cell, like proteases and other DNases, all these things which remain in the cell. So they are synthesized by this matrix ribosomes. And plasma membrane ribosomes are the proteins they transport to the outside. That means secretory. Okay, usually some extracellular proteins will be synthesized. So such kind of proteins are synthesized by this plasma membrane ribosomes. So domains of ribosomes. So within ribosomes, we have some domains. There are two domains. One is translational domain and exit domain. Translational domain means the region which is responsible for the translation. Translation is protein synthesis. So both subunits contribute to this domain. So we have a smaller subunit and a larger subunit. So I think I told you about them in the previous classes also. So smallest, the prokaryotes have 70s ribosomes, that is 30s smaller subunit and 50s larger subunit, and eukaryotes have 80s ribosomes, which are 60s ribosomes are larger subunit and 40s ribosomes are smaller subunit. And exit domain is the growing peptide chain emerges from the large subunit at the exit domain. So whenever this translation process starts, the two larger and smaller subunits will fuse at that point of time, and they will scan this mRNA for the code. And during in this process, the larger subunit, they consist of some sites, E site, P site, like that. So where this tRNA will carry the amino acid, whichever is there, which is complementary to the codon, and that will add to the growing polypeptide chain. And once the complete polypeptide chain is synthesized, that will be removed from the exit domain. So what are the dimensions of ribosomes? So in prokaryotic ribosomes, there are 70s ribosomes, and the size will be 14 to 15 nanometer by 20 nanometer. How they look like, no need to remember the dimensions, just remember the subunits. Okay, and a molecular weight is 2.7 million daltons. That much a molecular weight will be present, and they have larger subunit of 50s ribosome and smaller subunit of 30s ribosome. So how this looks like structure? So ribosomes are not membrane bound, and prokaryotic ribosomes are smaller and less dense than the eukaryotic ribosomes. And ribosomes are composed of two subunits, 50s and 30s, and they have proteins, ribosomal RNA also. So here we can see in the 50s, we have two RNAs, rRNAs, ribosomal RNAs, that is 23s and 5s, and it has 34 proteins. And in the 30s, we have 16s rRNA and 21 proteins. And the ribosomal subunits are like each subunit is constructed of 1 or 2 rRNA molecules and polypeptides. So this S, so in the previous classes also, some of you got doubt like it is not 30 plus 50 means it is 80. How we are telling 70? So all these things. So that is not regarding the like any number. So it is a Swedberg unit. So S in the 70S is and similar values, they are nothing but Swedberg units. So it is, that means whenever a centrifugation is performed, so usually we calculate the sedimentation coefficient. That means we will have the gradient maintenance. Okay, that means in the test tube or centrifuge tube, we maintain the different concentrations of the solution like sucrose or anything, and we will try to centrifuge by adding this sample of ribosomes maybe here, for example. So where it is settling, so according to that value we will give the Swedberg unit. The faster a particle travels when centrifuged, the greater is Swedberg value or sedimentation coefficient. So it is nothing but the particle's molecular weight, volume and shape. So heavier the component or particle, we will have the larger Swedberg numbers, or the sediment faster. They will settle down at the bottom very faster. So that's what, no, you can just by just for common sense, we can just think, if you take a stone which is very heavy, if you throw it in the water, so it will quickly go to the bottom and it will settle down. Whereas if you take any lightweight material and throw it in the water, it will be at the top or in the medium weight, so it will slowly goes to the bottom. So in the same way here also, heavier the molecular weight, it will quickly settle at the bottom so that it will have more Swedberg number. So this 30S subunit is the smaller subunit and it has a molecular weight of 0.9 into 10 power 6 Dalton. So it is having 16S rRNA and 21 polypeptide chains. So this is how, this is called base, this is called platform, and this is the head. Okay. Then 50S. So it's a larger one, has molecular weight of 1.8 into 10 power 6 Daltons. So it has 5S RNA, 23S rRNA, and 34 polypeptide chains. So here you have, these are called ridge, this is called stalk, and here you can see. And this is the back and this is the front. It is looking like a palm, right? And this is the central protuberance. So why ribosomal rRNA are present and what is the role? So rRNA is transcribed from certain portions of the DNA by the same energy requiring process used for the synthesis of mRNA and tRNA. So we have different types of RNAs. So the main ones are mRNA, tRNA and rRNA, but we have small nuclear RNA, small nucleolar RNA, small interfering RNA. Like that we have so many. So this rRNA has two roles. One is the 16S rRNA of 30S subunit. It helps in the initiation of protein synthesis. So they will have some sequences which will act as a signal for the initiation of the protein synthesis. And the 3' end of the 16S rRNA complex with an initiating signal site helps in the positioning of mRNA on the ribosome. So on this rRNA, usually any RNA or DNA will have 5' end and 3' end. So in the 16S rRNA, we have a 3' end where we will have a initiating signal site. That means it is a site where from where the translation starts. So this signal site, it helps the mRNA to correctly bind to the ribosome. And it binds to the initiation factor 3. So it has a signal CCA in the amino acid tRNA. So in the ribosome, we have A site, P site, and E site. So this ribosomes has binding sites for the tRNA. So first is P site. P site is peptidyl site or P site, where it is the donor site, and A site is the amino acid or acceptor site, and E site is the exit site. So here you can see this is the mRNA, and to the mRNA this 30 smaller and larger subunits will come and attach, and they will scan this mRNA, and they, whenever they start scanning, so the tRNA comes to this uh, amino acid site, and that will give the amino acids, and slowly that will be added to the growing polypeptide chain in the P site. So the growing polypeptide chains come from the P site, and once it is finished, so the tRNA will release, and this growing polypeptide chain will be released from the E site, exit site, and these two subunits will be dissociated. So what are the functions of the ribosomes? They are involved in protein synthesis. So this is again 3 steps like initiation, elongation, and termination. So in the real what you call the real process doesn't contain any initiation, elongation, or termination, but this is for our understanding, like we divide these components into 3 steps. So the initiation, the necessary components for the assembly are 2 ribosomal subunits, so the larger subunit and smaller subunit, and the tRNA with the anticodon UAC, and the mRNA molecule, and other additional protein factors. So these are the important components which are required for initiation. So in the E. coli and other bacteria, this begins especially from the modified amino acid tRNA, that is N-formyl methionine tRNA. So that is the modified amino acid tRNA. So here the alpha amino is blocked by the formyl group. This is used for only initiation. So this is called initiation tRNA. This is, it won't add any amino acid, but it is only for the initiation of the process. This N-formyl methionine tRNA attaches itself to the P site of the ribosome, peptidyl site. And the mRNA has a specific initiation codon, which is AUG. So this is universal. So anywhere the start codon will be AUG. So this binds with this modified F-met tRNA anticodon. Okay, and when this initiation signal starts, this 50S ribosome subunit will will bind to the 30S subunit and form the active ribosome mRNA complex. So this attachment is controlled by magnesium ions. So like this. So you can see this is the initiation codon AUG, and this tRNA, which is initiator tRNA, will come and attach it to this. So once it comes and attaches here, this will send the signal of initiation, and this larger subunit will come and bind to the smaller subunit. And now the complex is formed and it starts extending, elongation. The next step is elongation. So at the beginning of elongation cycle, the peptidyl site or P site is filled with the initiator tRNA already, and the amino acid A site with E site are empty. P site is already filled. Now this amino acid tRNA binding is done by, so when AUG is the start codon, and we have another codon which is adjacent to this. So the next codon is located at the with A site and is already to direct the binding of amino acid tRNA. So once the initiator tRNA comes and binds to the start site, the next codon is read already, and it will recruit the tRNA which is having the amino acid which is specific to that codon. And the GTP and elongation factors, they donate amino acid tRNAs to ribosomes. So the energy is required, right? So here, see AUG. So this first FMET is there, no? So that this tRNA is shifted to here, and another tRNA comes, and this growing chain will be added to this tRNA. Okay, then another tRNA will come. So the reaction which takes place here is transpeptidation reaction. So here the peptidase transferase, which is located in the 50S subunit, it catalyzes the transpeptidation reaction. So here the alpha amino group of A site amino acid attacks the alpha carboxyl group of the C terminal amino acid on the P site. Okay, so here you can see. So this is P side, this is A side. So the N terminal of here will attack the C terminal of this amino acid in this site. And it will try to form a peptide bond formation. So a specific adenine base seems to participate in catalyzing peptide bond formation. So like this. So you have, these are the amino acids. So this polypeptide chain will try to attach here by forming a peptide bond. Then translocation. So the movement of ribosome on mRNA is called as translocation. So there are three phases. The peptidyl tRNA moves from A site to P site, and the ribosome moves one codon along mRNA so that a new codon is positioned in the A site, and the empty tRNA leaves the P site from the exit site. Okay, and the translocation requires GTP and elongation factor complex. So this is how it looks like. So first new fresh tRNAs comes to the A site, and another tRNA with a growing polypeptide chain is present in the P site, and once this is done, so this tRNA moves here, and this tRNA along with the amino acid will attach to this growing polypeptide chain. So like this, it will move along the mRNA. Now finally the termination. So protein synthesis stops when the ribosomes reach one of the three nonsense codons, UAA, UAG or UGA. These are called stop codons. So any one, if it is encountered, the translation will stop. And once it stops, now this polypeptide chain has to be released. So that is done by releasing factors or release factors, which are 3 in number. So after the ribosomes has stopped, so peptidyl transferase hydrolyzes the peptide free from its tRNA, and the empty tRNA is released. And GTP hydrolysis is utilized in this process. And the next, ribosomes dissociate, that 50S and 30S will separate. And initiation factor 3 binds to the 30S subunit and prevent from the reassociation. Okay, only when the next translation comes, again it will start. So there are various antibiotics which can actually affect this translation process. So several antibiotics work by initiating protein synthesis on prokaryotic ribosomes like streptomycin, gentamicin. They attach to the 30S ribosomes and interfere. And erythromycin, chloramphenicol, they interfere with protein synthesis by attacking 50S ribosomes. Okay, so this is about ribosomes as well as protein synthesis also we have discussed. Let me share you just a second. Yeah. So this is about the ribosomes, like how the structure of ribosome looks like and how the protein synthesis also takes place. So we have also covered the translation process also, and we have again, I think in the next unit, but most of the process is covered. Now we discuss about the nucleus. So is it clear till here, like what is ribosome, what are lysosomes?
Yes,
Yes,
ma'am.
ma'am.
Okay. So now we will discuss about the nucleus, and then if time permits, today we discuss about chromosomes and all these things, so that it will be a basis for the next, like cell division, all these things. So what is the nucleus? So nucleus is the like headquarters of the cell. So it is having the complete genetic information of the cell. So hence it provides the entire information for the, like it is a blueprint for the cell. So in any eukaryotic cell, a large dark spot, it appears and it controls all the cell activities. So in the cell, you can see this is center one is the nucleus. So it is a membrane enclosed organelle which has genetic information and regulatory machinery. So it is surrounded by the nuclear membrane. So in nuclear pores are present. We have already discussed all these things. So what are the functions of the nucleus? So it stores the hereditary material, that is DNA. In case of other organisms, some have RNA, some have DNA, which is double stranded or single stranded, whatever it is, the genetic material is present in the nucleus. And it is a site of DNA replication and transcription to mRNA and ribosomal formation. So it takes place in the nucleolus. So the RNA and proteins are required for the ribosomal synthesis. They are present in the nucleolus. And it coordinates the cell's activity, which include growth, intermediary metabolism, protein synthesis, reproduction or cell division. So everything, gene expression, everything is regulated by the nucleus. So the structure includes that nucleus is enclosed by nuclear envelope. And the nucleus consists of chromatin, which is the genetic material. Then nucleoplasm or the nuclear matrix, like cytoplasm, we have the fluid filled cavity within the nucleus, which is called nucleoplasm. And nucleolus, it is the concentrated area of chromatin, RNA and proteins. So this is the structure. So here we have outer membrane and inner membrane. This together called as nuclear envelope. And inside we have nucleolus, which is surrounded by nucleoplasm. And here you have heterochromatin and euchromatin of the chromatin. It is of two types. So euchromatin means true chromatin, and hetero, which will be darkly stained, it will be tightly packed. And above that, they are associated with some ribosomes, and also nuclear pores are present. So this is how it looks like. This is under the microscope, how it looks like actually. So the nuclear envelope. So it completely encloses the nucleus and separates the cell's genetic material from the surrounding cytoplasm. So it is acting as a barrier, okay, so that the macromolecules cannot diffuse freely into the nucleoplasm and cytoplasm. So it is like a separation of nucleoplasm from the cytoplasm. And it has two cellular membranes, inner membrane and outer membrane. So they are parallel to each other, and they are separated by a space of 10 to 15 nanometers. And the outer nuclear membrane is continuous with the membrane of the rough endoplasmic reticulum. So this rough endoplasmic reticulum is having studded by the ribosomes. And the space between the membranes, outer membrane and inner membrane, is called as perinuclear space. This also is continuous with the lumen of the rough endoplasmic reticulum. So this is how it looks like. So this is nucleus, the bottom blue color one. So this is nuclear pore complex. This is lamina, intermembrane space. This is outer membrane. This is inner membrane. So in outer membrane are studded by the ribosomes here. And this even endoplasmic reticulum are present here. And this perinuclear space is in continuation with the lumen of the endoplasmic reticle, rough endoplasmic reticle. And the inner surface of the nuclear envelope is bound to a thin filamentous network. Okay, that is called lamins polypeptides or nuclear lamina. So this nuclear lamina provides mechanical support to the nuclear envelope and helps in the attachment of the chromatin fibers. And if there is any mutation in the genes which are coding for this lamins, then various diseases can occur like muscular dystrophy, so all these things. Okay. Then nuclear pore. So the nuclear pores are for the movement of RNAs and proteins. So everything which takes place inside that has to go out in the cytoplasm and from cytoplasm to nucleus. So that transport of RNAs and proteins is done by this nuclear pores. So the proteins which are synthesized in the cytoplasm, they cross this nuclear envelope to initiate replication. Okay, so even in the replication process also there are various enzymes which are involved. So that enzymes, all these things they come from the cytoplasm and they have to pass through this nuclear envelope. Similarly, this mRNA, tRNA, rRNA or subunits which are synthesized in the nucleus has to go out into the cytoplasm through nuclear pores. So the pore is 100 nanometer in diameter and around 100 proteins allow the free passage of small water soluble molecules, but preventing the larger molecules such as DNA and proteins cannot go. The larger proteins cannot go. And the nucleus of typical mammalian cells have 3000 to 4000 pores throughout this envelope. Now, what is a chromatin? So the interphase chromatin is, interphase means it is the gap between the G1, so that is between mitosis and the next cell cycle. So that is the gap, that is interphase chromosomes where the division is not taking place. So here they are highly extended nucleoprotein fibers called chromatin. And this chromatin is a complex of DNA, RNA and proteins. So these proteins can be histone proteins and non-histone proteins that make up the chromosomes. And each unreplicated chromosome consists of a single DNA molecule only. But when it comes to the mitotic chromosome, it is highly condensed. It is tightly packed. Okay. So here you can see. So this is the interphase chromatin which is freely distributed. Whereas when it comes to the mitosis, they will form chromosomes which are tightly packed and it will form a structure. So this is the karyotype. What we call as karyotype means it is a study of, it is a representation of the structure of the chromosomes and number of the chromosomes. So this is how the chromosome looks like. So we have a short arm and we have a long arms. Short arm is called as P arm and long arms are called Q arms. And the center portion where these two arms are located like fixed or attached is called centromere, and you will have a button-like structure which is called kinetochore. And these two, these pair of chromatids, they are called chromatids, will form chromosome. They are called sister chromatids. Yes, they look like similar. They are identical. Okay. So each human somatic cell consists of 23 pairs of different chromosomes. So in the diploid cells, and there are gametes which are haploid in cells. Haploid cells. What are gametes? Sperm and egg cells, which have only one set of the chromosomes. So one member of each pair is derived from the father and other member is from the mother. So one of the chromosome pairs consists of the sex chromosome, that is X and Y in case of females in humans, and X and Y in case of males, and X and X in case of females. This in humans. Okay. So in normal males, the sex chromosomes are Y chromosome inherited from the father and X chromosome inherited from the mother. So two X chromosomes are found in the normal females, one from each parent. And the other 22 pairs of chromosomes are called autosomes. And the members of each pair of autosomes are called homologs or homologous. That means they both are similar. Okay, because their DNA is very similar. But X and Y are not homologs because the DNA is not similar in X and Y. Even the size of the chromosomes also differs. So chromosomes are the physical carriers of the genetic information, and the packing of DNA into chromosomes involves several orders. So how the coiling and folding takes place. So what are the levels? Means first is the structure of chromatin varies as the cell progresses through the cell cycle. So the changes in structure are required to allow the DNA to be used and managed. So here, first the packing of DNA has to take place in a smaller volume to fit into the cell. This strengthens the DNA to allow mitosis and meiosis and also serve to control the gene expression mechanism. So see how this DNA is tightly packed and coiled in the form of chromosomes. So this is how it happens actually. This is DNA, and with the histone proteins, it will form nucleosomes and it will go on packing tightly, and finally after chromatin is packed, so now chromatids are formed. So this is how it looks like. So this is a simplified model of the DNA coiling. So first double helix, which is free form, then it will form nucleosomes, that is DNA with histone proteins, and this further tightly coiled to form chromatin fibers, and then they form chromosomes, and here it will form a loops, and finally in the metaphase chromosomes it is tightly and completely packed. Then nucleoplasm. So nucleoplasm is a highly viscous liquid that surrounds the chromosome and nucleoli. So many substances such as nucleotides and enzymes, they direct the activities that takes place in the nucleus. Okay. So this nucleotides and enzymes, they are dissolved in the nucleoplasm and they will participate in the DNA replication and other processes. So this nucleolus, next one, it's a prominent structure in the nucleus and it produces ribosomes which move out of the nucleus and takes position on the rough endoplasmic reticulum, which is critical for the protein synthesis, or some ribosomes may be freely distributed in the cytoplasm. So this is about the nucleus structure. And so now a little bit we will talk about the molecular organization of the chromosomes. So I already told you, like genetic material is nothing but which will like whenever a parent cell is divided into forming a new cells, so the genetic material which is present in the living cell has to be passed to the next generation. This is also called as hereditary material. And the bacterial genetic material, so this is mainly present in the nucleoid in the cytoplasm. Nucleus won't be present here. And the chromosome is single circular double-stranded DNA, which is attached to the plasma membrane. And histone proteins are absent here in bacteria. And E. coli DNA, it is circular and it has 4.6 million base pairs. And it has 4,288 protein coding genes, then rRNA operons, 86 tRNA genes. Okay, like that, it possesses everything within the nucleoid in the cytoplasm itself. So this is the bacterial cell. So where you have prokaryotic cell, you have plasmid. This bacterial cells or prokaryotic cells will have plasmid DNA along with the chromosomal DNA. So that's what, like, besides this chromosomal genetic material, they also contain small circular closed DNA molecules called plasmids. So they have these plasmids are like F plasmids, R plasmids, virulent plasmids, metabolic plasmids, which are not necessary for the survival, but they will perform some different activities in the body. Then virus genetic material can be DNA or RNA, and it is packed in the virus particle. Then eukaryotic genetic material, it is a genomic DNA which is present in the nucleus in the form of chromosomes. So now chromosomes. So a German biologist, Walter Fleming, so in 1880s, he revealed that during cell division, the nuclear material organized themselves into a visible structure called chromosomes. So these chromosomes can be identified by staining with the dyes. And the term chromosome was given by Walder in 1888. So chroma means colored, soma means body. So it's a colored body. That means whenever we can stain, we can easily identify. So it consists of many genes, regulatory elements, and nucleotide sequences. And chromosomes contains DNA bound proteins. So they are called histones. Okay, which help in the packing and controlling its functions. And these chromosomes, they vary in number and structure among the organisms. So for humans it is different, for other organisms it is different. So Benden and Boveri in 1887, so they reported that the number of chromosomes in each species is constant. That means in each species it is constant, it won't increase or decrease, but from one species to other species it differs. And Sutton and Boveri in 1902, they suggested that chromosomes are the physical structures which acts as a messenger of heredity. So these are the number of chromosomes which are present. You can see Arabidopsis thaliana. It's a plant model. So it has 10 chromosomes. It is diploid. And maize has 20. Like this is 2n is 10. That means 5 chromosomes. N is equal to 5 here. N is equal to 10 here. So like that we have in humans 46. So N is equal to 23. Right? Like that we have for every organism. So chromosomes are tightly coiled DNA and has basic histone proteins, which helps in tightly packing of the DNA. During interphase, they are not tightly coiled and they exist as chromatin. And the eukaryotes, so the DNA has to fit into the small nucleus. So therefore, it tightly packs, it undergoes condensation, and it is tightly packed in the form of chromosomes along with the proteins. So the shortest human chromosome consists of 4.6 into 10 power 7 base pairs. Okay, and this is equivalent to 14,000 micrometer of extended DNA. So that much of DNA is tightly packed here. So in the condensed state during mitosis, now this is about 2 micrometer long. See, 14,000 micrometer is packed to 2 micrometer. That means 7,000 times it has decreased. And now this DNA is packed stepwise so that this order of chromatin structure is called hierarchies of chromosomal organization. So this is the first level of package where the DNA around a protein, which is histone, to form a bead-like structure which is called nucleosome. And this structure is invariant in both the euchromatin and heterochromatin. And here 8 histone proteins, 2 H2A, 2 H2B, 2 H3, and 2 H4. And this center thread-like structure which is attaching this nucleosomes, that is H1. This is called linker protein. So these helps in the like packing of this DNA around this protein. So 146 base pair of DNA is wrapped around each nucleosome. And the second level is, once these nucleosomes are formed, now it becomes 30 nanometer. Now coiling of beads in a helical structure is called 30 nanometer fiber. That is found in both interphase chromatin and mitotic chromosomes also. So this further condenses. So this appears to be a solenoid structure with about 6 nucleosomes per turn. And that means every 1 micrometer along the axis, there are 40 micrometer of DNA is present. And this stability of the structure requires the presence of histone gene family H1. H1 is a linker protein. And now this is further tightly packed here. In the final level, the fiber is organized in loops, scaffolds, and domains that gives a final packing ratio of about 1000 in interphase chromosomes and 10,000 in the mitotic chromosomes. And here you have 700 nanometer structure, which is seen in the metaphase chromosome, and it is tightly packed to form the chromosomes. And extensive looping can be seen here. Okay, so we will stop here. So and we'll continue in the next class. So how the chromosome looks like in cell division and all these things. Okay, till here is it clear or any doubts are there? Yes, is it clear?
Why it is like only in the metaphase the chromosomes, ma'am? Before metaphase, we have even the prophase, no, ma'am. In the prophase, can't we see the chromosomes?
So that is initial stage. Like here, the separation of the chromatids, everything takes place, right? So complete separation takes place by the metaphase. You can see in the poles and all these things, right? In the prophase also you can see, but you cannot see them clearly, the separation and all these things, right?
Okay, ma'am.
In the metaphase they will be arranged at the middle and you can see, and is clearly they will get separated. Okay, so that's why we can see the condensation more clearly, the chromosomes in metaphase.
Yes, ma'am. Ma'am, then what about it is like a thread-like structure only, no, ma'am? That means there is no condensation in interface.
In interface there is no condensation.
That means there is no role of histone.
Sorry, your voice is breaking.
There is no role of histone.
No. In interface, there is no packing itself. So no histone proteins doesn't play important role there. Okay, once the cell division has starts, so in interface is like a resting phase. It's a preparatory phase, and you can the chromatin will be like freely dispersed in the nucleus, but only when the division starts, then they are tightly packed, and now the chromosomes has to get separated into the two daughter cells. So till that time there is no separation, right? So during cell division they has to get separated and they have to equally inherited to the daughter cells. So there the packing and condensation takes place.
Okay. Mama, you mentioned about sister. What is the difference between sister and non-sister chromatids, ma'am?
Means they are similar, like the DNA is like identical in both. Okay, so non-sister chromatid is chromatid of one chromosome and other chromosome. If you see, they won't look similar, right? So when we see, like, not all genes are present in a single chromosomes. For example, we have 23 pairs. So in the 23 chromosomes, all our genes will be distributed. So suppose the two chromatids of chromosome 1, they are similar. They are two copies. identical copies, hence they are sister chromatids. But the chromatid of chromosome 1 and chromatid of chromosome 2, they are not similar, they have different genes, so we cannot call them as sister chromatids because they are different. Is it clear?
Okay. Yes, ma'am. Clear, ma'am.
Okay.
Much.
Yeah. If any doubts are there, you can ask me in the next class also. And sorry, I will share the PPTs by today. So in the last week I couldn't share. Okay, so we will continue the class in the next week. Okay.
Thank you, ma'am.
If you don't have any doubts, I am stopping the class. Okay. Thank you so much.
Thank you, ma'am.
Yeah, thank you.